right. Okay, first of all, I guess maybe it's possible some of you have been looking for class notes from the last few classes and they're not uploaded yet. The reason is because somehow they're only letting me upload things if the file size is 5 megabytes or less. And I have a lot of pictures and even movies and things like that in my PowerPoint, and they tend to be very big. It's not easy to make it smaller, so I'm working on kind of reducing the file size and making it more amenable to uploading. So please be a little patient, and I'll try to upload lecture 4, 5, and 6 sometime the rest of the week. We'll also next week have a short quiz over what we've gone over so far. That was going to be part of this class. It's not going to be very big, just 10 simple questions. That should be just so I can kind of gauge where everybody is and how much learning is going on here. Okay. And so last time, we talked about chromosomes in the nucleus, and I ended up with talking about how connections between chromosomes and the cell work. So, for instance, the centromeres can act as handles that the microtubule cytoskeleton grabs onto. And I finished by giving a kind of sneak preview of some other type of cell-chromosome interaction that happens in the process of meiosis that involves not the centromeres, not the center of chromosomes, but the telomeres. I'm going to get back to that quite a bit from now, but before we get into meiosis, I thought it was a good idea to kind of step back and approach the whole study of chromosome biology from a different but equally important historical perspective. So far, I've kind of emphasized the cell biology aspect of chromosomes, so using microscopy, how people discover chromosomes inside cells with stains and then use microscopes to look at them and see how they behave. But at the same time, there is another science developing, the science of genetics, that ended up, it started in a very different place, but these two fields kind of ended up converging in the modern field that we know now as modern cellular molecular genetics, but we can say that chromosomes are kind of at the center of that. So I'll go over the basic introduction to genetics, and if you already know the material, hopefully you'll at least learn some English terms. If you already know both the material and the English terms, then this will be a nice review for you. But we'll start at the very beginning. Before there was anything called genetics, there were people who crossed plants together and observed what they got. And people have been doing that, of course, for thousands of years. And noticing that, of course, when you cross different plants or animals together, the offspring sometimes look like the parents. That was common knowledge for thousands of years. But this gentleman here, named Gregor Mendel, started to actually count the numbers and count in a quantitative sense just how much did these progeny look like their parents. And it was very simple experiments that he did, but it ended up really changing the world because he discovered how heredity really works. Okay, so in a typical example, and this is a cross that he really did. What I'm drawing here are peas. It's just some kind of mame, just peas, and that's what he worked with. And he had different kinds of peas, and these were actually the seeds that he was looking at. So there are some peas that had seeds that were yellow and round, very smooth, and then other peas had seeds that were green and very wrinkled, so kind of not very smooth, but shiwa. And he noticed that if he started with plants that were always yellow and round seeds, so it came from a long line of peas that were yellow and round, and you never saw any green or wrinkled peas, and he had another plant that always had green and wrinkled seeds. When he crossed those two plants together, they actually didn't look like both parents. They would only look like one parent. So all of the offspring, all the progeny of that cross, when you cross them together, had yellow and round seeds. And it didn't matter which way you crossed them. So peas and all plants, they have male and female flowers. And it didn't matter if this was the male flower and this was the female flower, or vice versa. It was the same result. That when you cross these two kind of plants together, all the progeny meaning the next generation, had yellow and round seeds. And so because of this, the yellow and round trait of these seeds, he called these are dominant, because the yellow color is dominating the green color, and the smooth round surface is dominating the wrinkled surface. Okay, and as a complement of the word dominant, there's the word recessive, which means even though one parent had these two traits, these two qualities, it did not show up in the next generation. So to summarize this, if you had plants that were always yellow and round, so their previous generations had yellow and round seeds, and that previous generation had yellow round all the way back, and the same with the green and wrinkled plants, then they would give rise to these other plants that are yellow and round all the time. But of course he didn't stop there. In fact, I think everybody at the time probably knew that. But then, if you keep going and take these seeds and do another cross with them, so take these seeds and then cross them back to this one parent, then what you saw is if you take these yellow and round seeds and cross it to this green and wrinkled plant, then instead of having all yellow and round, then you would get a mixture of green, wrinkled, yellow, and round, all different kind of seeds. So apparently there is some difference here, right? So this parent, even though this was a yellow and round plant, and this parent, which is also has yellow and round seeds, there's something different about them. There's something different about the plant that always has yellow and round versus a plant if you have just crossed it to a plant that's green and wrinkled. And so we can say that there's a difference here. There's some plants are yellow and round and they always have been, whereas other ones are yellow, but maybe they have some bit of hidden green in them. They're carrying some green trait, but it's hiding, it's not visible. And same with this wrinkled trait. So somehow by crossing it to this green and wrinkled plant, they picked up this extra bit of information, of this element, but they're not showing it here. So it's hidden in this generation, but it comes back when you cross it to this generation. So he had the idea that in addition to this yellow and round traits that were visible and the green and wrinkled traits that were visible, you could also have green and wrinkled traits that were hidden, that were not showing up. And when he counted how many of these different kinds of offspring you got, what he saw was that you actually had equal numbers of each possible kind of offspring. So if you counted how many yellow and round there were, how many green and round, how many green and wrinkled, how many yellow and wrinkled, there were about the same number of each one. And then he kind of thought about what would have to be the case for this to work. So if you cross this the yellow with the hidden green and a round with a hidden wrinkle versus you cross that to something with only green and wrinkled, and you've got equal numbers. Well, what he came up with was this. So imagine this is the plant that has just wrinkled and green traits, and that's all it has. And this is a plant that has at least a yellow trait and a round trait, but then there's also some hidden green and wrinkled trait in it. And then this is the final outcome. These are just example numbers where if you count about 100 seeds, you would end up with about equal numbers of each. So he realized that, so if you make an assumption that in addition to this yellow and round trait, you simply got an additional green and wrinkled trait that's exactly the same, just hiding in here. And then each plant gives some, and we can also just label them with letters so it's a little easier to see. And each plant gives one of their traits to the pollen or to the flower that they're going to produce, and then these unified again, then that just explained everything. So in other words, in each little pollen made by the male plant, it picks one of these, either the hidden one or the expressed one, from the left side, and also one from the right side, and puts it into a pollen. And the one that's always wrinkled and green has only this kind of pollen, so it doesn't really, there's no variation there. So all the variation comes from one parent, and if you just make the assumption that each of these elements exists, the expressed element and its hidden copy are equally real, and each one can go to the seed, then it explains the kind of numbers that he saw. Okay. And just for equality, he also made the assumption that the one that was always green and wrinkled, it also had 2 copies of each, because there's no reason to, because the cross worked both ways. So it just gave him this model that that for each trait, yellow or wrinkled, there was 2 versions, and each plant always had both versions. But when you go to the next generation, only one of each version goes to the next generation. And he called this his first law of segregation. So he came up with these, what he called the laws or rules of heredity. And the first one was this, just that although each genetic element, color or smoothness, it exists in 2 copies, 2 kinds in each plant, but when they go to the next generation, only one can go into a pollen or into the flower. So you have to pick yellow or green, and you have to pick smooth or wrinkled, and that was his law of segregation. Okay, so that's what explained the idea that you could have this hidden trait. He didn't really go into why it was hidden. He just assumed that one version was a strong version and one was a weak version, and the strong version was always the one that showed up. Okay, and so this is. So this, if you look at, use a kind of more modern symbols for these traits, the dominant traits, the ones that show up that are not hidden, we usually write with a capital letter, and the ones that are hidden, or that can potentially be hidden, are written with a lowercase letter. So usually a gene that gives rise to a dominant trait is usually written with a large letter, and vice versa. Gene that often gives rise to a recessive trait is written with a small letter. And you may note that I'm not saying a dominant gene or a dominant allele or a recessive gene or a recessive allele, because the genes themselves are not dominant or recessive. It's actually the trait that is dominant or recessive. So in his mind, it kind of looked like this. So each, he didn't really have an idea of what these elements were or how they were organized. But all he knew was that each one existed in 2 copies. And when they made their pollen, those copies came apart somehow. There was no idea of how they came apart or what they were. He didn't even know if they had physical form. He didn't necessarily believe that these were physical objects, only that they were 2 elements, right? They didn't really know much about atoms or molecules or how life worked at all. So really, all he could say were that there were these genetic elements or elements of heredity that had this sort of structure. So sometimes they were together, sometimes they came apart, and then sometimes they got back together. Okay, but he didn't just come up with this theoretical model. He was very thorough. So if his idea was correct, that each trait had 2 copies, then he should be able to predict the outcome of many more crosses. He should be able to predict, for instance, if you started with this kind of plant that had one capital and one small of each trait, and crossed it to itself, so just took one plant and crossed it to itself, you would think he should be able to predict how many of each seed he would see in those progeny. And he predicted it very, very well. So the numbers that he got matched very well with the prediction of his model, which is that each each, say the female and the male, can give only one of each trait to their offspring. And you're probably familiar with this way of writing these crosses. This is called a Punnett square, and it works as long as you have one or two of these traits. Once you start to have three, it gets a little too big. But this is a good teaching tool so you can see what all the combinations add up to. And so if you have, right, if his model is correct, that you have only one of each trait going into the seed, then if you go down this column, everything has a big Y and a big R from the pollen. So that's these two. And for instance, here everything has a big Y and a big R from the female flower, and so that would be these two. So and if you assume that the numbers are equal, so that there are as many big Y's as big R's and as many small Y's as small R's, then this is how these numbers work out. So the only time you would get a green and wrinkled seed is if you happen to pick the small y from both parents and the small r from both parents. And since you have a 1 in 4 chance of this happening and a 1 in 4 chance of this happening, then you have about a 1 in 16 chance of that happening. And the numbers there would just be kind of example numbers from if he counted 1,600 different seeds, but whatever number he decided to count, the ratio would always kind of end up like this, in this really nice 9 to 3 to 3 to 1 ratio. So what that is if you have two parents and each parent is what's called, I should tell you, this term is heterozygous. So this would be, it has a capital and a small together. So that's heterozygous. And the other term, homozygous, would be if you have both the same. So either that or this. Okay. I'm not sure he actually came up with these terms. It might have been added later. But heterozygous means you have two different versions of one of these elements. Homozygous means that both of your versions are the same. Okay, and if you're heterozygous for two different traits, then this is the ratio you would expect to get. But you should, this math works out so beautifully like this, only if you assume that this, the Y trait and the R trait have nothing to do with each other. That the R does not know what Y is doing and Y does not know what R is doing. In other words, it has kind of this structure in mind, that once these things are segregated, the Y's and the R's are completely independent and they don't know what they're doing. They don't know what the other one is doing. And only under those circumstances do you get this, this 9 to 3 to 3 to 1 ratio. And so because of that, because he saw this ratio so many times, he decided that this would be the second law, that this is the law of independent assortment, which means that if you have this trait and this trait, that these will go to the seeds randomly, and then these will go to the seeds randomly, and they don't know what each other is doing. Okay. So that also explained the fact that he got equal numbers of each of these, because so the number of, right, yeah, because otherwise, yeah, each of these 4 elements has an equal chance of ending up in any of those seeds. So they're independent, and that's why you would get equal numbers. But it didn't take long for people to find some exceptions to this. So this law of heredity is more like an ideal law, and we can state that it's not that it's not that the elements are independent of each other, but that they often are independent of each other, or they can be independent of each other. But in many cases, they're actually not. And when they're not independent of each other, then you get cases where these numbers are not necessarily equal. Okay, so for instance, it wouldn't be these same yellow and wrinkle, because those are actually independent, and that's how we discover them. But let's just say pea plants have many different characteristics, but these are really easy to draw. So let's change. Let's just assume for a minute that this is a different pea plant, maybe where these things are not independent. So instead of counting equal numbers of each pea plant, you could easily do something like this, right? Where if you cross this plant, which is double heterozygous, with this plant, which is double homozygous for the recessive versions, then this is a pattern that you would see. This is a possible pattern that you would see. In other words, you would see a lot of yellow smooth seeds and a lot of green wrinkled seeds, but then you would not see any green smooth seeds, or you would not see any yellow wrinkled seeds. And so what this means is obviously these Y and R elements are not going independently of each other anymore. So the capital Y seems to be linked to the capital R, because you get those together, but you do not have capital Y next to a small r. All right. So that led to a slight modification, which is instead of Y and R being independent of each other, they're often linked together. So in some cases, they can be linked together, and so they will travel together as a single unit. So instead of a plant choosing randomly this one versus, so a male plant will still choose randomly this versus this, but if they choose this one, if they choose the capital Y, then they will have to choose the capital R as well. They cannot choose capital Y and a small r. So in other words, if you were to count these seeds and see this kind of pattern, then you would know that these genes are grouped together somehow. And so he got about this far and then decided that it was too complicated, and it was kind of disappointing, I think, that not all the genes worked out this way. But it turned out to be actually a great discovery, because if it weren't for this, then genetics as we know it just wouldn't work. So this was also fairly easy to understand. It was accommodated by the theory pretty easily. Okay, so either Y and R being independent, unlinked, ungrouped from each other, that's pretty easy to understand. These two elements just don't know what the other one is doing. Alternatively, the two elements being linked together, that's also easy to understand. So maybe color and smoothness somehow always go as one unit together, and that's also pretty easy to understand. But what was really difficult to understand, and took people a long time to accept was really happening, was observations like this, where instead of this complete linkage, instead of yellow and round being completely linked together, you would actually see occasionally a yellow and wrinkled seed or a green and smooth seed. But it's not equal numbers anymore, and it's not zero, but instead it's some small number. But almost all the time, the small number is the same. So in other words, you have just about as much chance of separating the big Y from the big R and getting big Y, small r, as you do separating getting the big R and the small y. So this and these two classes were always equal to each other, but also not equal to these guys. So in other words, what it looked like was happening was, so instead of them always being linked together, what you have is a situation where they're linked to something. These elements are linked to something, and that something can sometimes change, can sometimes break and join apart, break and rejoin in a different way. So this is the model that then made sense of this sort of observation. So normally, this big Y and J R, they're linked together to some kind of substrate, some kind of scaffold, and they usually travel together. But sometimes that scaffold can break and rejoin with the other one. And this would explain why these numbers are always the same, because again, it's this scaffold itself that is assorting independently into the seeds and not these genetic elements. And that was kind of the first clue that these elements are probably physically real. Like these elements of heredity are things that are connected to some physical object. And the more people studied this, and the more people studied not only 2 traits at a time, but 3 or 4 or 5 traits at the same time, they would see this kind of linkage between traits happen very often. And not only could multiple traits be linked to each other, but the numbers started really making sense after a while, and people started making very careful counts of these sorts of crosses. So for instance, if we introduce another trait, let's call it P, which will be, again, its color, but let's say it's the color of the pod that the seeds come in, and not the color of the seed itself. And that can also change independently of the seed color, because it's a different time in development. So if you made this sort of cross, for instance, you would have to first make sure that you had some plant that was always green and wrinkled, and I think the pea pod actually does go the other way, so this is the recessive and that's the dominant. A little bit confusing, but green seed, yellow pod, and wrinkled skin. And you would breed this for a long time, so you would know that it's homozygous for all those traits. And then you would have another one that you made from a cross that you knew was heterozygous from all these traits. And then you would count. If you counted the offspring, very often you would get something like this. So what's called the parental types would be the same traits as were in one of the parents. So capital R, capital Y, capital P would be exactly reproducing what the female parent had, for instance. You could also get what's called a single crossover, which means this substrate here, which we're leaving. I'm leaving ambiguous what that substrate is for now. I should tell you that this is what people refer to this as a crossover, because this substrate made this cross shape if this were to happen. And that's going to be a word we use a lot in the future. So. So if you have this kind of event where the scaffold breaks and rejoins and it separates the parental traits from each other, then that's what's called a crossover. And so if you start with the parental these parental versions, but then switch to this recessive version, then this it just made one crossover right here. Or if you start with this big Y and then have RP, then that can also be explained by one single crossover. And these were fairly common events, but you'll note that the crossovers in this region seem to be less common than the crossovers in this region, because you more often have these two together. These two are more often together than the capital Y and small r. Okay. And then there are some kind of combinations where you would have a yellow seed and a wrinkled seed, but then a green pod. Those were the most rare, and less than 1 in 100 times. But actually, if you multiply these two together, this is what you would get. And it was this could be explained by having 2 crossovers at the same time, and that was also observed with some rarity. And the most simple explanation for this type of difference why would you more often get a crossover in this region than in this region? Can anybody think of why that might be? Why might you be more likely for these two traits? Yeah.
No.
Yeah. Yes. I think you're right. So exactly. So it's longer. That region is longer, and so the crossover itself can be random, can happen anywhere. But this, if you adjusted these lengths to accommodate these frequencies, then it made sense. And it really made sense because once you started adding more and more genes, I just said genes, but yes, of course, that's what these are. These genetic elements are genes. And you assign, instead of these frequencies, you assign a length unit called centimorgans, which is pretty much the same thing. It's how many out of 100 do you see. Then eventually you get these really, really complicated maps. But the beautiful thing here is that all of these distances work out. So eventually it was realized that these scaffolds that these genes are sitting on are these linear elements. So it was experiments like this, counting the different kinds of offspring and colors and shapes of offspring, that made people realize that these traits existed as two copies on these linear structures that could undergo these crossing over events. And the length was such that if you measured the distance between these two and you measured the distance between these two, it would more or less sum up to the difference between these two. So if you ignored this, if you ignored the wrinkled trait and the round trait and only focused on y and p, then you would come up with a number that was 20 out of 100. So that all made sense. And so at the same time people were figuring this out, or actually a little bit before people were figuring this out, you had these guys who I think I've introduced, or anyway, I've introduced their contemporaries. Right, I don't think I've shown you these guys before. But these were typical early 20th century cell biologists who were looking at all the different kinds of cells they could and looking at chromosomes with microscopes like this, so probably straining their vision a lot. But they made really detailed observations. And they were probably looking at organisms with very small numbers of chromosomes. And they were particularly interested in the germ line, so in the cells that the cells that are involved in going to the next generation, so sperm and eggs. And what they saw, and what was highlighted in Walter Sutton's manuscript here, I don't know if you can read this, but so before I get to that, what they noticed was that very often the number of chromosomes they would see in a species would undergo this alternation of in generations. So that, for instance, if they saw 10 chromosomes in the somatic cells, in the body cells, then they would see 5 chromosomes in the sperm or the eggs. And then when the sperm and egg came together, it would go back to 10. But for another organism where they saw 24 chromosomes to begin with, it would have 24 chromosomes in the body, then only 12 in the sperm or the egg, and then back to 24 when the two came together. And that was really a mystery to them until they got a hold of Mendel's work, and they started thinking about these genetic elements that Mendel had described and how there were 2 of each of them, and how they came apart from each other, and how they came back together when an egg was made. So in 1902, this guy is writing that the probability that this association of paternal and maternal chromosomes in pairs and their separation during the reducing division, and that's an important word, as indicated above, this may constitute the physical basis of the Mendelian law of heredity. So, and he says, I hope to soon return to this subject in another place. But yeah, this was an extremely important connection that people made between these physical things that they could see with their eyes that were doing this doubling and halving and doubling and halving with generations, and these purely theoretical entities that Mendel had described that also did the same sort of thing, where there were 2, and then 1, and then 2, and then 1. So this connection really made people focus on chromosomes and put a lot of effort into proving this this theory, which became known as the chromosome theory of inheritance. And it was kind of, as hard as it was to think of that theory in the first place, it required a lot of creativity and insight to think of that theory, but then it took a lot of hard work to actually prove it beyond the shadow of a doubt. And we'll probably talk more about that in a later class. But for now, what they did manage to find out, or to put these two, to put the cell biological observations together with genetics, they came up with these conclusions: that chromosomes are actually the genetic material, or the material of heredity. So all the traits we've been talking about, they somehow exist on chromosomes, or in fact they are chromosomes. And then the segregation of these maternal and paternal chromosomes to opposite daughter cells is exactly the same as the segregation of factors that Mendel saw with his pea plants. And the independent assortment of genes, so when you have yellow seed color going independently of the smoothness of the seed, that's because they're physically located in different spaces. So these two traits, they're not actually overlapping each other. They're not the same thing. There are two different things that occupy different locations. And what we saw with all these genetic mapping experiments was that they occupy these linear positions. So they occupy specific positions on chromosomes, and you could predict the outcome of a cross if you knew the positions of these genes on chromosomes. So that was the conclusion of proving this chromosome theory of inheritance. This is what people started out with, and a lot of experiments were done to prove this. But one of the most elegant pieces of evidence for this is not any kind of experiment, but just looking at one form the chromosomes take during this process. So remember, there was this postulated process of crossing over, where the scaffold that these elements are attached to would break and then come together again. And by looking at the right kinds of chromosomes, people could actually just see this exact thing happening. So precisely the kind of physical arrangement of this genetic material that had to be true for you to get these multiple crossing over between scaffolds could be seen in chromosomes if you look the right way. This is an electron micrograph, so this is much later than the actual evidence for this theory. But even with old-style light microscopy, people could see things like this. And so this is actually one chromosome that is crossing over to another chromosome. So it's picking up whatever trait was on this chromosome, and then going back to the other parent, and picking up this trait. So in the end, it's going to have whatever was here, and whatever was here, whatever was here, and whatever was here. And that's why we don't look exactly like our parents, which is probably a very good thing. Okay, so that kind of is the genetic background for the following bit, which is going to be more about the actual cell division of meiosis. Now I'll be using terms like homozygous and heterozygous and crossover that we've had before, I think. Meiosis is, as you know, gensu bunmetsu. It's this reductive division. So the guy I showed you just a while ago, when he was writing about this, he didn't have the word meiosis, but he said the reducing division. And that's more kind of direct translation of the Japanese term, which is really like reductive division. That's what meiosis is. It's a cell division that's specialized to take one genome, however many chromosomes are in that genome, and end up with another genome that is half that number. So this is the most simple example, of course. This is a cell with one pair of chromosomes, and then in its sperm, instead of a pair of chromosomes, instead of 2 chromosomes, there's just 1 chromosome. So being able to reduce the chromosome number by half is extremely important, and it's what meiosis is specialized for. Because, of course, when the sperm and the egg come together again, you are doubling the number of chromosomes in that cell. So if you didn't reduce the number of chromosomes, then each time sperm and egg met, you would just double and double and double the genome. So of course it's essential that you first cut the genome in half in order to make the sperm and eggs in the first place. And the way you actually get 4 sperm out of this single precursor cell is that you have 2 divisions in a row with only 1 round of DNA replication. So what I haven't showed you earlier is step 0 here, where each of these chromosomes replicates first. So this one chromosome is actually made of 2 sister chromatids held together by cohesin, as you remember. But this is the chromosome that came from the father, for instance, and this is the chromosome that came from the mother. And each one is replicated. And so it's this first division, which is the unique cell division in meiosis. Because, as you will note, remember earlier I mentioned in mitosis, when you divide chromosomes in mitosis, of course this cohesion comes apart and the sister chromatids go to opposite cells. But you'll note that's not happening in the first division of meiosis. Instead, the maternal and the paternal chromosomes first separate without getting rid of their cohesion. And that's very important, and that's how we can reduce the number of chromosomes from 2 to 1. And then the second meiosis division is pretty equivalent to just a normal mitotic division. And that's when the cohesion actually comes apart in the end. Okay. And of course, as I mentioned before, that it's a very important cell division. And its importance comes from the fact that it's easy to mess up. And in fact, for humans, it's especially easy to mess up. And we have, and in fact, the oocyte division in particular can go wrong very, very, very often, and that leads to the chromosomal abnormalities and infertility syndrome that I've talked about before. This increases rapidly with maternal age. I keep hitting on this, but it's quite important for our health, that around age 40 or so is when the percentage of this kind of aberrant cell division starts to increase. It increases somewhat in men as well with age, but it's harder to do the oocyte, the female cell division, than it is to do the male cell division. There have been some pretty exciting recent work that shows how a lot of it is due to the cell size. And maybe I'll be talking about that in future classes. So it's an important question of what can lead to these kind of failures, and to think about what can happen to get a failure in chromosome segregation. We need to know, first of all, how does it happen? How does chromosome segregation in this first meiotic division happen in a normal case? So when everything goes right, what has to happen in order for the first cell division to occur? So there are a few steps that always happen regardless of the species and regardless of male versus female. So meiosis always starts with pairs of chromosomes that are independent from each other. They're not touching, with very few exceptions. Paternal chromosome 1 has no idea where maternal chromosome 1 is. Remember the kind of genome organization that I showed you earlier? So how chromosomes are arranged in the nucleus. Sure, they're in their chromosome territories, but maybe paternal chromosome 1 is over here and maternal chromosome 1 is over here. And these two chromosomes need to end up in different cells. But at the beginning of meiosis, they have no knowledge of each other, and there's no connection between them. So the first thing that has to happen is that those chromosomes need to find each other. Somehow chromosome 1 needs to find chromosome 1, chromosome 2 needs to find chromosome 2, etc. They find each other and meet up along their entire lengths And they undergo this process called synapsis, where this protein structure links them together. We'll talk a little bit more about that one later. And it's in that context, it's when they're lined up together. So the maternal and paternal chromosomes are lined up together that they can undergo this kind of crossover event and do this. So now what started off as purely paternal chromosome 1 now has the left end of paternal chromosome 1 joined to the right end of maternal chromosome 1. And you can have multiple crossovers. But they have to first be really, really close together, not just kind of touching on their sides, but all the DNA needs to be really close together because for this to happen, the DNA sequence has to be right identical, right? Because you can often cross over in the middle of one gene, and you wouldn't want to cross over the wrong way. So you wouldn't want to have an extra A in here because that could lead to a mutation. So the genes have to be precisely lined up, and this process of synapsis, which can take hours or even days, is what lines up chromosomes so that they can undergo this recombination. And then after this recombination, then chromosomes are able to pull apart from each other. And if they don't recombine, chromosomes fail to recombine, then they won't be able to come apart. But let's just talk about the first part because to me it's the most interesting because these chromosomes start off not knowing where each other is, not knowing where their partner is, and it's kind of like this problem we face all the time, right? You know that there's two of each in there somewhere, but for meiosis to succeed is kind of like expecting these socks to find their partner's sock inside this pile somehow, all by themselves, without being able to look. So there's little cellular machines, there'd be little machines in there putting the socks together in the right way. But that's how meiosis works. So the question is, how do chromosomes know which one is their partner? And so somehow they go from this unpaired configuration to a configuration where they all have met their partner and they're all happily paired with each other. So just to think about how hard of a problem that is, it's actually much harder than it seems. You know, we all do our laundry, so we don't think it's that hard to pair up things. But we can start to just use some numbers to get a glimpse of how hard this is. So let's start with a very simple organism. Not the simplest ant with only one pair of chromosomes, because that's a little too easy. But let's say if you have just two chromosome pairs. So this flower has chromosome 1 and chromosome 2. So all their genes are on either chromosome 1 or chromosome 2. And it gets, just like we do, this flower got one of each chromosome from a male flower and one of each chromosome from a female flower. And then in the body cells, or in the vegetative cells, they have two of each. So so here are the chromosome pairs, and somehow this red chromosome has to pair with that red chromosome. This blue chromosome has to pair with this blue chromosome. And how many ways are there to actually pair the chromosomes? There's actually 3 different ways to do it. There's the correct way, and then there's 2 incorrect ways. Right? So this is correct, this red pairing with this red, but the dark red can pair with the light blue, the dark red can pair with the dark blue. And then these possibilities are just forced based on this possibility. So there's 3 different ways to pair up these chromosomes, and only one of which is correct. So this flower has a 1 in 3 chance. If you just say, okay, I'm going to pair my chromosomes together, there's a 1 in 3 chance that it will succeed. Okay, well, that's not too hard. So if you make 30,000 pollen grains, 10,000 of them will be correct, and maybe that's good enough, right? Well, okay, what if you have, instead of 2 chromosome pairs, what if you're a slightly different kind of flower and you have 3 chromosome pairs instead? Well, now if you consider again the single chromosome, it has 5 partners to choose from. And once it's chosen one of those 5 partners, now there's 4 chromosomes left, and now you're back to the situation of the previous guy where there were 3. So there's 5 initial choices, and then there's 3 second choices. So now there's 15 ways to pair your chromosomes together. And again, only one of these ways is correct. All the other ones are incorrect. This will give you a functional egg or functional pollen. All these will lead to sterility. And of course, you need 2 of these correct, so it's actually even harder. Okay. And now, what I usually do is ask the class how many possibilities are there with 4 pairs, and just see who is getting this answer right. Anybody? See if I remember.
Is it 4 factorial?
It's very close. Is that times 3 times 2? No. No, so this one is already 5 times 3. But you're close. I mean, yeah, it's definitely a factorial progression here. But it's actually maybe a tiny bit faster. So the formula is this. So if n is the number of pairs of chromosomes, then it's 2n factorial over n factorial times 2 to the nth power. Okay. It's actually not that hard. It's simply you double this and then multiply all the odd numbers that lead up to that. So again, I'll ask you how many possibilities are there? Can you raise your hand if you know the answer? It's okay if you don't want to shout it out. You know.
Yeah, let's say 5.
Yeah. Yes. So right. So if you have 4 pairs of chromosomes like this crocus, that's right. There's 105 different ways of pairing these chromosomes together because now there's 8 total chromosomes. The first chromosome has 7 partners to choose from. That leaves 6 chromosomes left, so now it's just like the previous one that had 15 choices, which really has 5 choices, and then it's like the previous one that had 3. So each additional pair of chromosomes puts you one more odd number up in this progression. So when we think about humans now, and we have 23 pairs of chromosomes, this is how many different ways there are for us to randomly, if we were just saying, okay, just zip up, just whatever chromosome you're closest to, just pair with and go from there. You have 1 in this many chance of getting it correct, which is essentially 0. And of course, if you are that monster fern that I talked about earlier, sorry about the margins there. I guess the font is too big. But anyway, yeah, there's just astronomically high. You don't get numbers this high in too many, even in too many physics. But yeah, that's how many ways of pairing these chromosomes you would have. But if you look at this is actually a meiotic cell where all 631 chromosomes exist in pairs. So this is one chromosome paired to another one. This is one chromosome paired to another one. They're doing crossing over. So somehow this fern was able to pair all those chromosomes together. Okay, Right, it's not, I've made it seem impossible, but actually it's not, since we're all doing it, there obviously must be some way that chromosomes are brought together. And if you think about it as this random process, of course it could never happen. And if chromosomes just had to assort randomly until they found their partner, then you would wait forever for that to happen. So there are non-random processes or ways that chromosomes exploit the kind of order or organization inside the cell that they can use to make this process easier. And one really important way that they do this is this organization inside the nucleus that I showed you last time, which is this bouquet configuration, where all the chromosome ends come together at one part of this nuclear envelope, and the rest of the chromosome is kind of put out. Here's all the chromosome ends here, and you can see that they're all kind of sticking out here. So what's thought is that these chromosomes right here are moving around very quickly, and they can, since they're all pointing the right way, they're already predisposed to at least be able to compare the right part of themselves. So, for instance, all the left ends of all the chromosomes are here, as well as all the right ends. So if one right end hits the other right end of the correct chromosome, then that chromosome will pair up like this, and it actually makes this protein scaffold that kind of takes it out of the equation. So every time you get a successful pairing event, that pair of chromosomes is removed from that equation. So as time goes on, a few chromosomes correctly pair, and then the problem gets simplified and simplified. So as the more chromosomes are randomly hitting their correct partner, they make the problem easier and easier until you only have a few chromosomes left. So I think a lot of what makes it easy is this progressive nature of how pairing continues. But unfortunately, a lot of this is still a mystery, so we still don't know what exactly it is about chromosomes that lets them recognize their partner. Maybe it's a little disappointing. I don't have the answer, but it's really an active area of research. So the basic question is, how does your chromosome 1 from your father pair with chromosome 1 from your mother and not chromosome 2? How does it recognize, oh yes, this is chromosome 1 and that's my partner, and chromosome 2 is not? All we know is that, for instance, the DNA sequence, of course, is different. They have different DNA sequences, they have different lengths, different packing densities along the chromosome, but we don't know which of those is the most important. Eventually it comes down to DNA sequence, but there are also a lot of DNA sequences along our chromosomes that are exactly the same. We have these transposons and viruses and other things in our genome that make our genome look very similar from one part to another. So why doesn't that mess up this pairing process? It's still kind of unknown, that's why I like studying it, because it's still one of the mysteries in biology. And the problems don't end there. Okay, so not only do we have not only is this combinatorial problem pretty hard, but remember I said that this is an incorrect view of chromosome organization in the nucleus. It is for interphase and for mitosis for all of our body cells, but if we were to look at a cell undergoing meiosis, undergoing pairing, so when chromosomes are looking for each other, they really do look like that. Okay, so they really do start here, and they have all this kind of like spaghetti-like organization inside the nucleus. You can tell here that a lot of it kind of has this bouquet configuration because many of the ends are down here, and you can see areas where two chromosomes have actually found each other. So when they're really close to each other like this, that means that they're finished, and these are removed from the equation. So now this chromosome is not going to be looking anymore. Maybe these two are starting to find each other. This one seems to have not found his partner yet at all. So this is a nucleus that's trapped in the act of finding its partners. And so if you remember, one of the reasons one of the likely advantages of not having this kind of organization in interphase is that chromosomes don't get tangled up with each other. But in this case, they really do. Okay, so chromosomes in meiosis not only have this problem of finding their partner, but they have this tangling problem, which can be seen here. And I think I'll turn the light off so you can get a better look at this. So this is a picture I took about maybe 9 years ago. These chromosomes are from corn or maize, and the protein that we're looking at is REC8, which is actually one of the cohesin proteins. So it's one of the proteins that goes all along the axis of the chromosome, holding the sister chromatids together. And because this is a protein that goes along the axis of sister chromatids, when the two homologs, when the mother and the father chromosomes come together, there's two of them. There are two of these axes, and that's why it looks like this double line. And you can see that these chromosomes that I'm showing you in this three-dimensional movie, they're pretty much finished, right? They're completely lined up with each other all along the lengths until you get to here. And I hope you can see that this one chromosome here has this opening. Do you see this bubble here? And what's going on, this is the one place in the entire nucleus where the chromosome pairing has not finished. And what's going on here is that this other chromosome is actually going through it. So the chromosome wants to finish pairing, but this other chromosome is in the way, and it's not allowing that to happen. So this is actually kind of a rate-limiting step. This resolution of these entanglements inside this nucleus is one of the last things that has to happen, because we know in the end, all of this is finished and the chromosomes are all separate and not tangled together anymore. We still don't know how these chromosomes are able to resolve this, but most likely it involves some kind of unthreading process where one chromosome simply is pulled out from this. So that is, that's probably how an interlock, this is called an interlock, that's probably how this kind of interlock can be resolved. But there's another kind of interlock that can't be resolved that way, and that's when you have some situation where two chromosomes are actually pairing. I'm going to draw it correctly. Let's see. Two holes. Yeah. So you can also see configurations like this. Where this is just zooming in on one region, where two chromosomes have paired almost completely, and they're almost finished, except they've encountered this sort of entanglement. And you can see this is not a simple matter of being able to pull one through the other. This is really like a knot in these two strings. And we see this just as often as that other kind. And yet, somehow, they're able to resolve this. So either this connection has to come apart somehow, or the chromosome has to break, and we don't know which one is happening right now. Yeah, this is kind of just showing, this is all about the mysteries of meiosis. Another thing that makes this difficult, of course, is that if the maternal and paternal chromosomes start off on opposite sides of the nucleus, then in order to pair with each other, they of course need to first move. They need to undergo some sort of motion, like somehow going throughout the nuclear volume, searching for their partner until they find the correct one. We think probably a lot of movement can be imparted by this zipping up process. So chromosomes come together and they start to zip up, and that itself is making chromosomes move a little bit. But for these first contacts to happen, like those contacts at the nuclear envelope, they need to move there in the first place. Let's see. I guess, yeah, I don't think I have any movies of them moving, unfortunately. I'll try to get that in there next time. I guess what I have here is instead some different examples of this sort of interlocking. So like the kind that I just drew would be this one. Yeah. So this is the kind of an actual picture of this sort of interlock that will prevent pairing from finishing. And in fact, you can get really, really complex tangles like this, where you can have 4 or 5 different chromosomes all making a single knot in the middle. And it's really not clear how the cell will ever be able to deal with this. But yeah, somehow they manage in the end. Yeah, so that's a very good example of these 2 bubbles, as they're called. Yeah, but they're all resolved in the end. And okay, that's pretty much what I wanted to get through today. The only other thing I will show you is maybe introduce what I'll be talking about next time, which is the system I'm using now. I don't work on corn or maize anymore to look at meiosis. Even though the chromosomes are really nice and big and beautiful, and it's really nice to work with under the microscope, this C. elegans, this nematode, has great many advantages in genetics and being able to actually do experiments on them. These make a new generation of worms every 3 days, so you can do crosses and genetic manipulations much more quickly than with maize. C. elegans have 6 pairs of chromosomes, so it's very easy to count. And if you see 6 chromosomes at the end, then you know that meiosis succeeded. If you see more than 6 or a different number than 6, then you know that something has gone wrong. So it's very easy to see whether meiosis has succeeded or not. And one thing that makes it so nice is that half of the worm's body is devoted to producing meiotic cells. So the gonad of the worm takes up half of its body. And you can really just see inside it. The body is transparent, so you can see these meiosis cells inside very easily. And so looking at it to see whether meiosis has succeeded or not is really easy to do. Okay. The other really nice advantage is that within this gonad, the gonad is a long kind of tubular organ inside the worm. Meiosis is proceeding in this kind of spatio-temporal pattern, so you can observe meiosis happening in a single animal at many different stages. And just one example of that is looking at, for instance, chromosome 5. You can see it pairing over time by marking one region of it with this in situ hybridization technique, where you use fluorescent DNA to mark a single part of the chromosome. And if the chromosomes are not paired, you will see 2 dots, because each chromosome 5 will have its own dots. So you can see this nucleus, those 2 dots are apart, these dots are apart, and they're apart. So this region of the gonad is starting meiosis, but the chromosomes have not paired yet. But as you continue, and this is only a few hours later in time, and just a few tens of microns in space, then each chromosome 5 has found its partner and successfully paired. So with cytological assays like this and genetic tools that have been developed for the past 40 years, the C. elegans is a very good system to study this. And so, yeah, I think I will introduce more about C. elegans and some of the work I'm doing next time. And I will have a movie of chromosomes actually moving. I'm sorry I didn't include it this time, but it's really nice, and it's a movie of in C. elegans. So I think I will stop there and